Your daily experience of vision feels immediate and complete. You open your eyes and receive a seamless, high-fidelity stream of reality. Biological hardware tells a different story. To see an object, light must travel through the eye's crystalline lens and undergo refraction. This lens relies on structural crystalline proteins to flex and bend parallel light rays, forcing them to converge onto a fixed two-dimensional focal plane at the back of the eye. This diagram visualizes a massive systemic bottleneck. The retina contains roughly 100 million light-sensitive input points, yet the optic nerve exiting the eye contains only 1 million output channels, forcing a 100-to-1 data compression. This physical routing introduces a second constraint, neural latency. It takes tens to hundreds of milliseconds for compressed data to navigate these pathways and reach the brain. The visual system bypasses these hardware limits by fragmenting optical data into parallel processing channels, which the brain then reassembles via high-frequency neural synchronization. The brain builds conscious vision through time-staggered data reconstruction, synthesizing fragmented signals into a stable internal model. This reconstruction begins at the front of the eye, which operates as a tunable convex lens. Ciliary muscles constantly adjust the lens's curvature to maintain an exact focal length based on the distance of an object. Because this hardware consists of biological tissue, it degrades. Crystalline proteins lose elasticity with age or lose transparency through cataracts. High-fidelity initial signal capture is therefore critical for the system's survival. Light projected onto the retinal wall lands directly on the outer segments of the photoreceptor cells. This technical model illustrates the microscopic architecture of these cells. Focusing on disc morphogenesis, we see the mechanics required to maximize photon capture. The ciliary membrane evaginates, folding outward, forming nascent disc lamellae, stacking them into a dense column. This stacked geometry maximizes the surface area exposed to incoming light, sharpening the signal-to-noise ratio at the exact moment of photon impact. This high-fidelity capture ensures vital data survives the impending 100-fold compression waiting at the optic nerve. The photoreceptor hardware mathematically optimizes initial sampling to feed the highest quality data into the neural circuitry. Once the signal becomes electrical, the retina shifts from transmitting raw pixels to parallel feature extraction. This schematic highlights retinal ganglion cells, or RGCs, at the bottom, acting as final exit nodes. Dozens of subtypes divide incoming light into independent variables, running distinct channels for motion, color, and spatial detail. This timeline illustrates how these parallel channels introduce asynchronous arrival times. Data for motion and edges fire down the nerve rapidly, reaching the brain in about 40 milliseconds. Information regarding sustained luminance lags behind, taking up to 240 milliseconds. Fast dynamic features arrive out of sync with slow static ones. The brain receives a moving edge long before it receives the object's surface lighting. This staggered delivery enables a predictive coding strategy. The brain calculates the trajectory of urgent threats immediately, prioritizing motion while waiting for the rest of the image to arrive. The retina solves the compression paradox by parsing the environment into asynchronous, pre-calculated features. This creates an integration problem. The visual cortex must somehow reassemble staggered fragments of edges and colors into a single object. This feed-forward input routes from the optic nerve through the lateral geniculate nucleus and into the primary visual cortex. Empirical recordings provide the answer. Looking at these coherence maps, we see evidence of gamma band synchronization. Immediately following a stimulus, localized bursts of high-frequency electrical activity emerge, centered in the 30 to 80 Hz range. Integration occurs in two phases. First, fast retinal oscillations between 60 and 120 Hz drive the early visual cortex into a state of rhythmic timing. Second, as the feed-forward wave arrives, intrinsic cortical networks generate a slower 30 to 60 Hz rhythm. These rhythms act as temporal glue. By firing on the exact same millisecond interval, neurons processing different features bind themselves together across the layers of the cortex. Precise rhythmic timing unifies these staggered signals. The data integrates only when these distributed neural clusters fire in perfect temporal lockstep. The successful execution of gamma band synchronization directly determines the threshold of conscious visual perception. When synchronization is weak or disrupted, parallel feature channels remain disjointed, and the perception of a unified object fails to emerge. This oscillatory wave physically stitches together disparate representations, facial recognition, grid orientation, and motion detection, to produce a single state of unified awareness. This dynamic binding acts as an error correction mechanism, aligning raw sensory input with top-down cortical predictions. The system's stability relies on three structural pillars. First, the crystalline lens and stacked photoreceptors that gather the initial physical signal. Second, the retinal circuitry that compresses that signal into stagger parallel feature spikes. And finally, the cortical rhythms that bind those asynchronous fragments back together. The immediate, flawless reality we experience is a masterful hallucination built across multiple physiological scales to overcome the inherent limits of biology.